on to what everybody is here for, our wonderful speaker. So, as you will be aware from the description, Dr. Steve Kassem is a neuroscientist, works at Neura, and he will be talking to us about brain mapping and whether it's possible to have Google Maps for the brain. So welcome, Dr. Steve.
Thank you. Now, let me just get this over to the screen. All right, fantastic. So I haven't embellished these slides very much. I hope you don't mind. Blank slides, I'm sure, are more helpful for challenging me. I'm going to walk through a few things. So I'm going to give you a bit of history. I'm going to have a lot of bold statements, which I know skeptics like to challenge, and I'm going to give you the information you need to be able to challenge me on those. There'll be a few philosophical statements and a lot of biases from myself. So just to quickly run you through a few things, I've got a PhD, I'm a member of the Royal Society, and then there's all my other credentials there. But to start, now we know this, but just to emphasize it for everyone, the brain is the most important organ in any body, not just our body. In every organism, all organs have evolved, and all of evolution has evolved to support the brain to facilitate survival. That's everything. Your liver exists by virtue, direct or indirect, to support the brain. Your heart, your lungs, and everything else is there to give the brain everything it needs. For humans, it's evolved to the point where it can give us our thoughts, our emotions, our movements. Love comes from there. Everything comes from there. There's nothing you can conceive of, past, present, or in the future, that can manifest from anywhere except the neurons in your brain. Now, unlike other organs, we've only been fascinated with the brain for about 6,000 years. So even before Aristotle, now he did have an opinion on the brain, and we're going to get to that in a minute, and I thought we'd all appreciate a little bit of phrenology here on the screen. I was actually challenged by a Nature reviewer recently that the work I was doing on mapping the brain was akin to phrenology. And I want to see if you can see how that might be the case by the time we get to the end. Because there's some bold statements we make with brain mapping, which is that every single part of the brain can conceivably be mapped. By virtue of that, that means, once you figure out that, you'll see the numbers when we get to the end, so it's quite unlikely to get there, but should we get there, that means there should be no thoughts you have that we cannot then map. And that has various ethical implications if it were to be true. But let's start historically. So the first use of the brain comes from the Egyptian manuscripts translated from Edwin Smith's significant works, where they have a word for brain, principally referring to the organ inside the skull from Egyptian warriors who have had injuries to the skull. But since that point in time in Egypt, we get to Aristotle, who sort of had the boldest statement regarding the brain. Before that, the brain was sort of considered either an air conditioning system to keep the body cool, waste organ, all sorts of it. But Aristotle took it a little further and argued that, well, at least it's the first surviving account, which has argued that it is organised and it's significant, and that he organised it into 3 parts. But something I need to preface before we jump into regional neuroanatomy, which is I suppose the more formal term for brain mapping, is that it can often be considered, even within scientific fields, to be rather inconsequential. What does it matter that we know that there's a hippocampus that organises our memory? I mean, of course, yes, people can appreciate that memory comes from the hippocampus. But why is that important to my gene I'm studying, or my particular cell type I'm studying? Well, all mapping and all neuroanatomy is fundamental to all of neuroscience. And if I haven't talked myself up enough, that means it's fundamental to everything we do. So even if you can't appreciate the organisation of the brain and how it's mapped and how it's connected at face value, you should be able to at least appreciate how your cells or how your genes or however any particular minutiae that you work in science might exist within a neural system, and from that you should be able to yield more information. Now, from Aristotle, things didn't really change much up until Vesalius, the very significant anatomist, who in his manuscript De Humani Corpus Fabrica, or On the Fabric of the Human Body, was one of the first major anatomical works that has survived, where he used direct observation, which hadn't actually been used since Aristotle, to cut open bodies, identify organs, map them out, identify them, and try to justify each of their existences. He too couldn't really make a justification beyond the significance of the brain needing to exist for human behavior and life, but couldn't really take it much further than the three-part system that Aristotle proposed. He did extrapolate on the existence of nerves, but I put all this together to give you this image here, taken from his book. This, as you'll see, if you can keep this in your mind as we go through the talk, isn't that different to where we are now. On this image, you'll see letters identifying different anatomical features in the brain. You've got A through to G, covering dura mater, gyri, sulci, veins, sinuses, and the rest. And we still, more or less, from a macroscopic level, define the brain in these broad structures. And that's because we haven't really developed a means of taking it much further than that. So this is the first modern representation of the brain, and it is still more or less the same representation of the brain we have now. We do have some improvements. We have Franz Nissl and Aloysius Alzheimer, who used the Nissl stain, one of the first significant histological stains, and that's where we use chemicals, we dip tissue into those chemicals, and those chemicals react with certain properties of the cells to reveal certain features of those cells. So what you see on the screen here is a sagittal cut. So that's where we put the brain into the deli slicer, ear first, and slice this way. And this is taken from the center of the brain if we were to do a sagittal cut. The dark blue, purple, I'm colorblind, not the field of science for me, I know. The dark blue purple is cortex, that's the density of neurons, brain cells, and the more fainter areas, the opaquer areas, are white matter, that's your connections. And so using this histological stain, we could see that there's a cortical ribbon, we could see many of the anatomical features of the brain that we could not see otherwise, just by cutting it open using Vesalius's methods. And from this stain, beyond the anatomy that we could appreciate, we were able to identify some things like Alzheimer's disease, where the cortical ribbon was thinner in people that had what we were calling at the time senility. When since then, those that died without senility didn't have the same thinning, and we could identify that as a disorder. And so that's how we were able to get Alzheimer's disease. But Franz Nissl also, as many neuroscientists were, just before the turn into the 20th century, quite philosophical about these new discoveries about the brain. Understanding that we had these neurons, and then understanding that should these neurons be damaged in a particular disease or a particular way, that changed this person's behavior. Now this is post hoc, we're understanding after the person's death we can determine these features. But he had a rather bold statement. Now it's German, and I translated this in my PhD rather poorly with a German dictionary because Google Translate was awful at the time, or didn't exist really to any useful degree. But the translation that he originally used, as best as I could have done it at the time, was: as soon as we agree to see in all mental derangements the clinical expression of definite disease processes in the cortex, we remove the obstacles that make impossible agreement among alienists. Now, if we were to put this in a more modern frame and paraphrase it a little bit, the statement he's actually saying is all irregular behavioral phenotypes must be expressed as irregular changes in neuronal morphology. So his statement is that any change in behavior from normality must be explained neuroscientifically. There is no way. Should you like the color red? That's because your neurons make you like the color red. Should you like eating Caesar salad that's gone cold? That's because you're happy to tolerate that because your neurons have arranged itself in such a way. And then this can be extrapolated to disease states, psychiatric states, anything else that you can take from that. This is also an extrapolation that he made from a common game that used to be called Socrates Questions, if anyone knows this game, which is you can play a game of ad nauseum questions to explain any behavior in any person. This morning, I had coffee. why did I choose to have coffee this morning? Well, because I have it every day. Well, why do I have it every single day? Well, because I've gotten addicted to caffeine, and without it I feel lethargic. So why did you get addicted to caffeine? And then, I'm not going to play this game ad nauseum for you. No one actually likes it. Socrates was killed for a reason. But you can play this game ad nauseum until you get to a point where you cannot do anything else but agree that the behavior you had was caused by something outside of your free will. Now, this is something often forgotten, not exactly the Socrates question, but the understanding of behavior being a representation of neurology is often forgotten in the fields of psychology, where the mind still exists. Now, just to get us up to modern, because we don't want to talk about history for the entire talk, we land at Brodmann and his Brodmann areas. Now, anyone who's done any psychiatry, psychology, or neuroscience, this is where we're at. He used a combination of behavioural analysis, surgery, and those histological stains I was talking about to create a modern map of the brain. On this image, taken from his original works, he uses a combination of colours, shading, and numbers to distinguish the 47 parts of the cortex. Each of these 47 parts of the cortex are still what we use to map brains now. Your neurosurgeon would use Brodmann's maps from 100 years ago, unchanged, to map the brain still and understand where a tumour might be or a particular behaviour might be from a result of damage or tumour. Now, despite the tone I'm using to talk about these are actually supremely useful and were done extraordinarily well and have been repeated ad nauseum to get a very refined map that represents our behaviour. Now, it does seem a bit reductionist to bring all of our thoughts and movements down to 47 parts of the brain, and that's because it is. At the moment, you'll see that there are about at least 1,000 parts of the brain that we've identified in modern work that are not represented in medical work. They are purely in scientific work. And because it is difficult to absorb 1,000 regions of the brain as a medical practitioner, you can't absorb all of them. But these 47 give you a fantastic tool to utilise in the medical space. Now, a quick little piece of trivia, because no one just wants facts all the time, is the Nissl stain that we use, that Franz Nissl used in the late 1800s, is actually derived from the violet flower. He crushed violet flowers and a few other heavy metals together to get a stain that was able to bind to the Nissl bodies of our neuronal cells, and with that, give us the colours that we see in the brain here. So we have another Nissl stain of the human brain here, coronally cut this time. So this is if you put your nose in the deli slicer and we cut through the whole thing. And we've all had dinner already, so it's okay. And again, you can see this lovely patination in the brain. I find it quite artistic, that you can see these beautiful striations, and each variation of colour you see is arguably a different part of the brain that has a different responsibility to different thoughts, actions, and motivations that we have. Now, there are obviously many histological stains as well. Here's just a few more. We used 8 in our most recent atlas that we used, and we used 8 because each one of these stains will stain for a different chemical feature or biological feature, and then we can superimpose each of these stains on top of each other to sort of play a Venn diagram game of trying to find more structures that mightn't necessarily be identifiable with only one histological stain. And so, for example, there's Nissl, ACHE, parvalbumin, and so on. But histology is not the only tool we can use to investigate the brain. We've come a lot further since needing to do investigations post-mortem. We can do them in vivo now, in the living person, and we can use MRI. So, I mean, we've all probably been exposed to an MRI in some way, a magnetic resonance image. And what you see here is an MRI of the brainstem, highlighted in red on the spinning graphic on the right. Sorry, speaker angle. On the right, and then from that we have these different contrasts that we can use. Now, contrasts are similar to histological stains, but in the MRI space. So just like we could use Nissl and Parvalbumin and all those other colours you saw to stain for different chemical features, we can use different contrasts of an MRI to reveal different features as well. Now these contrasts, we can talk about the physics of them a little bit, but it's essentially playing with the magnetic field within the MRI to reveal different physical features of the brain. And what you see here actually is the highest resolution image of a brainstem in the world. So the work we do in the Paxilon's Brain Mapping Lab is the highest resolution in the world. So our maps are the most used in the world. It is a demountable with an 82-year-old Greek man who works 100 hours a day, myself, second in command, and then a few other PhDs underneath me. And we do better than our chief competitor in the US, which is the Allen Brain Institute, which is a $200 million institute with 100 postdocs. So, and so what you're seeing here is a 50 micron resolution image of a structure that is about 5 centimeters long. So 50 microns, that's 0.05 millimeter steps in each pixel. And then on the far right over here, you have a tractographic image. This is where we try to trace each of the pathways that would be running through each of these voxels, these pixels, on the image to try and understand where the neurons are connecting themselves to other parts of the brain. So each one of those fibers represents about 10,000 neuronal connections. Now, this image here is taken from a living person. This is some of our more recent work where this is the highest resolution image of a living person's entire brain. It is 200 microns, so a fifth of a millimeter per resolution pixel. And the color information you see on this image gives you directional information. So that tractographic image we were looking at before, that's a tiny bit of guesswork. We have to use mathematics to sort of guess with high probability where these pathways are going. But in this situation, the colors represent physical direction. Red being left to right in the brain, green being in and out of the brain, so front to back, and blue being up and down. And color combinations of those give you the appropriate combination of direction. And so I get to a point halfway through my talk here where I want to give you the fact that got me into neuroscience. And also the biggest challenge probably in mapping the brain. I've given you a lot of confidence, we're doing very well, we're the best in the world, we've got all these lovely images. But this is the hurdle. The brain is not just the most important organ in our body, but definitionally the most complex noun in our language. There is approximately 80 to 100 billion neurons in the brain. Each of those neurons sends out at least one dendrite, on average 5, with each of those dendrites synapsing to another 100,000 neurons. A synapse is a connection in this context. It is these connections between our neurons that give us our thoughts and emotions and gives us our consciousness. The higher the degree of connections, the higher the degree of intelligence could be ascribed to a particular central nervous system of an animal. So what you can do though is you can calculate the possible number of permutations of these connections between these cells in our brain, and the number becomes quite large quite quickly because you're going to 100 billion to the power of 100,000, then times to the power of 99,999, and so on. To give you a context of how large this number is, I want you to think of the amount of atoms in the tip of a pin. It's about 10 million. Then the amount of atoms in a grain of sand, it's about 400 billion. Then think of how many atoms there are to constitute our entire planet, then constitute our solar system, then constitute our galaxy, and that number is still too small by comparison. The number of possible combinations in our brain's connections is actually larger than the number of atoms in the universe. So you have on your shoulders something that is mathematically possibly more complex than the universe you exist within. Now look, there's a tiny bit of grain of salt here. I'm obviously talking a little bit of fanfare here. But we haven't mapped every connection in the brain. So it is still possible, until we map all the permutations of all the connections in the brain, that the possible number is larger than the number of atoms in the universe. At worst, though, it is at least the number of atoms in the Milky Way. So you have at least that much complexity in you. Now, if mathematics was not enough of an inspiration for you, it's a bit faded out, but that's an MRI with gyrifications on it. Let's see if I can artificially increase my brightness. Let's see. That won't do anything. Well,
You trust it.
trust me. You can see it. We've got an MRI of the brain there. We've seen that. We've seen what halfway through the brain looks like. Can someone tell me where this image is from? It is very obviously. Thank you. It is God, drawn by Michelangelo. Now, if you had a little bit more contrast and brightness, you would see that the cherubs' shapes, their shoulders, the gaps between the cherubs, and where God's legs and hands are placed, perfectly marry the macroscopic anatomy of the sagittal cut through the brain. You would argue, now you'd believe me more if you can actually see it. I'm not playing a psychic game here. Richard can see on my screen that it does match. It is arguable that Michelangelo, who was a bit of an anatomist, well, at least appreciated anatomical drawings, was trying to give us a little hint to say that God isn't God, but it is in fact in our skulls. It is brain. It is the brain that is God. Now, if you like this type of concept, not a person I'm a friend with, just a fan of a very good book. There is a fantastic book that elaborates on this a little bit more called The Executive Brain. Now, just to impress you a bit more, this is one of my favourite images I've made myself. This is generated from the highest resolution image that you saw earlier, doing a tractogram again, as we saw before, but of the entire brain now, not just of the brainstem, which is that small blue bit at the bottom. And as it spins around at this resolution, to render it so it's under a gigabyte big, each one of those strands you can see represents about 100,000 connections, or 100,000 pathways, I should say. And again, the colour is giving you the directionality of that connection, blue being up and down, green being front to back, and red being left to right. It's quite lovely, even as a colourblind person, so it must look even better to you. Now, just to give you a little bit of context, I'm becoming a bit unbelievable at this point. So this is our lab, there is our group, there's me on the right there when I was a little skinnier, and we have our professor in the middle, our PhD student on the top right, and then 3 of my postdocs underneath. So this is the Paxonos lab, George Paxonos in the middle at the back there. He's worked so much from a contract he had from 1973 that the federal government had to step in to force him to use his 6 years of annual leave he accrued because he'd never taken a day off because he loves this job too much. He sued the government too for forcing him to take the leave. But what you can see here are 2 of our most significant books. We do many other atlases. You have Atlas of the Human Brain, the most used atlas of the human brain in the medical and science space, used by surgeons, used by researchers, and everyone in between. And then you have the Rat Brain in Stereotaxic Coordinates, which is the most cited book in Australia, the most cited neuroscientific book in the world, and the 3rd most cited book of all books in history, at 150,000 citations. Every single scientist who's ever studied the rat brain, done a psychological, psychiatric, or neuroscientific study, has likely used the rat brain in stereotaxic coordinates. Now, what makes the mapping we do so special is that we do it by hand with tracing paper, compasses, and triangles, and a magnifying glass. As you can see up there, we have a light box illuminating through photographs that we have on top, and we have all of us sitting around literally tracing and identifying everything we see. Beyond the joy of doing that and being able to be paid to do things like that, the result is a map that is refined. It isn't just that we've traced it, is that every pencil mark has to be agreed by all the doctors at the table that what we're seeing is what we're seeing. Then this gets refined again and again and again and again. Every quarter millimetre or every 1/5 of a millimetre in the most recent atlas we're doing, we do this exact same image, trace it all again, and if it doesn't match up, we have to do it again to find out why it's not matching up, and repeat the process ad nauseum. Each atlas takes about 5 to 7 years to make. Now, it's not just the accuracy, it's not just the comprehensiveness, I should say, of these maps that make them so useful. It is also that they're stereotaxic. That means, in the simplest sense, that they are translatable to any other brain. You can look at these maps and go 3 millimeters across, 4 millimeters back, 6 millimeters down is this structure. And then you can use your brain with very little mathematical ratios, get to the exact same location and the exact same spot in the brain. And that's what makes it the most useful for surgeons, because I've got a big head, literally and figuratively, and other people don't have the exact same size. So you need a bit of ratio on this stereotaxic space to be able to get to the exact spot. And just because we had, George had a photographer come in on his 50th anniversary of working full-time, take some photos, I thought I'd share them with you too, because they're quite stylistic. Now, this is ones I took, just showing you in the lab. So we lay out our lab, all the flat surfaces, not just the benches, but any surface that can hold paper. We lay them out flat with our photographs. And this way it gives us a very panoramic view to appreciate it, but also gives us something that a computer screen cannot do, which is visualize all the screens at once. I can look at an entire centimeter of slices with 100 slices in there and look at them all at once and being able to see any error almost instantly, because we have a very good skill at doing that. A computer, though, I would have to change screens repeatedly, and I would have forgotten about the 0.5 millimeter difference that I would have had on the previous image by the time I would have got to the next one. And so it sounds a little silly, but it is actually one of the most important features to some of the work we do. Just to give you a little bit of context again, so this is a slice through the brainstem again, horizontal slice. So sagittal through deli slicer in the ear, coronal deli slicer in the nose, horizontal deli slicer from the top of the head down into the brainstem. Now this, although it's aesthetic to look at, you can see some of the patinations of the Nissl stain through this brainstem, and you can probably identify the snake of patterns down there, that's the olive, and some other clumps. We turn that into this, where each one of those labels identifies a unique structure that has a unique function in the brain, that has a unique outcome to the performance that we have in our behavior or thoughts. I'm not going to run you through all of them, obviously, but we have the photograph of the labels on top, and then we have a diagram that people can use a little bit more freely to be able to orient themselves. And again, you see those stereotaxic coordinates on the sides to be able to orient yourself. But histology is an older field, and as much as George wants to stay in that space, I'm trying to drag him into the MRI space. And again, just because these are pretty pictures, this is another image through the brain of another participant that we were able to convince to sit on the MRI scanner for 24 hours. Now, that participant couldn't be found, so I volunteered, and with the help of a Valium, I was able to get it done, and we were able to get this beautiful picture from a living person. And from that, just like with the skill of histology, and in the exact same way of printing them all out, overlaying them all and drawing on all of them, we're able to identify the structures you see there on the left-hand side. And again, turn them into a diagram that lists all the structures on the right-hand side and everything else someone might need, with the red line identifying where the brain slice has been taken. Now, principally, what I've been working on recently is a pediatric brain atlas. So what shocked me recently was that pediatric MRI brain atlases don't really exist. Mind you, there is very few post-mortem ones as well. Oftentimes, we are forced to extrapolate from the adult. Now, this isn't exclusively the case. We're not saying that pediatricians have nothing at all, but I was surprised to learn, talking to the director of the Sydney Children's Hospital, that almost always they must extrapolate from an adult map where things are in the child, let alone in a 15-year-old teenager, a 5-year-old child, or an infant. It's all extrapolation. Now, this is a combination of difficulty in obviously trying to scan a child, and no one, ethically, you're not permitted to donate a child, as disgusting as that sounds, for post-mortem analysis. So there has been a lot of ethical constraints. It's not from desire that we haven't had this, but the new MRI techniques we're getting means that within 7 minutes, we're able to scan a child, which is a lot easier to keep still than 24 hours. But just to give you some context of why this is a big problem, because extrapolation's not too bad, and obviously there hasn't been too many significant problems, but it creates a circular logic which I can only assume has biased our work over the decades, which is that by extrapolating from the adult, by using, in fact I say it better here, by using adult brain anatomy as a framework to study developmental changes in the child brain, it presupposes what it aims to discover. You're already aware of what you're planning to find, so it makes the idea of even doing so redundant. Now, we've only just started, so this is some pilot work. The Haskins Atlas on the left is the best in the world up until our work. That is their MRI on the left with the map that they superimpose over the top. We've picked the area where they identify the most structures in a single image, which is 16. Our pilot data, which is significantly better than that, you'll see some of our fresh off the press last week stuff on the next slide. But this is what we've mapped so far, has 60 in the same space. And this is significantly different enough from the adult to just justify a grant we recently received to investigate this further. Now, this is, I really wish the contrast and the resolution of this projector was better, but they're just going to be useless. But appreciate with a bit of imagination that what we've got on the left there was what we showed in the pilot data, and then our recent work on the right here is where we're at the moment, which is the best in the world. Though I want to give you skeptics a tiny bit of a help here to help attack me later in the questions, which I'm looking forward to, which is that we have used AI in a most recent imposition from our directors, because directors love to throw AI at everything, to improve our brain images. Now it has, but it's not a raw image anymore. The AI has technically invented pixels to smooth the image out and make it better. Now, has that directly impacted or lied to us? It's hard to say yet, but I've got a student working on a comparison between raw images and AI-assisted images in MRI brain scans to look into this further. At the moment, it looks like it's not causing any problems, but I look forward to it showing something, because who likes AI in anything? Now, lastly, I want to show you where we're moving towards, which is... Oh, really? Oh, well, I suppose that's what happens in every talk when you try and show a video. One of them doesn't work. But appreciate that what we're getting to the point now is we can make an entire brain transparent, make the cells illuminate in a particular color, have those cells feed back through light waves to give us information, so that we can get the benefit of both histology and MRI in a single scan. We get the advantage of histology, of single cell understanding and analysis, of seeing how single cells react to certain chemical compounds, while also being able to get full 3D representation of the brain because it's transparent. I'm very sorry the video doesn't work. It is very stunning. Now, just some final thoughts before I let you go, which is the study of neuroanatomy, again, with a bit of history. I appreciate history in this space, which we start with Nicholas Steno, another famous anatomist, who said that anatomy should not be bound by any rule, but ought to change as frequently as dissections have begun. His argument was, with the complexity of anatomy, every time you open it, with direct observation, you should be able to find something new if you look hard enough. Now, that was coming from 1669, so there's a little bit of bias there. However, it is still the case that we are discovering new parts of the brain now. Now, what do you mean? Surely we've cut the brain open enough and we've seen all the structures inside. No, we've just got to look with new technology and new tools, and we were able to find a pea-sized structure in the brain of the human called the endorestiform nucleus. Endo meaning within, restiform meaning rope, and nucleus meaning clump of cells, within the human brainstem. This hadn't been identified before, and it was able to be identified recently. So that's on his side of the argument. However, Herrick, a more modern neuroscientist, argues that the terminology of the brain is in great confusion. And this is actually the case, because with all of these new tools and with all of neuroscience exploding as it has, what has happened is each lab has developed its own idiosyncratic mapping system for each of the new parts of the brain they've discovered. Each new pathway gets a new brain, which means that when you try to read someone's neuroanatomical paper from a different lab, somehow they've got the same name as you, but it's a different part of the brain, or they've used non-formal terminology, or up is down, left is right, who knows? And that was the problem in 1915, and it has only made itself worse as of today. But the question I'm always left with as a person who maps the brain is, when all parts of the brain are eventually mapped, all of them, more than the 1,000 we have at the moment, when every single neuron is mapped to understand every single function it has in the brain and how it influences our thoughts and how each of these pathways form circuits to justify behaviours and movements down to minutiae of how my hand is wiggling the microphone at the moment would have something slightly different to when I am swaying my body. Can there be any room left in the brain for free will when we can map every single action and thought you have, and then by playing a game of Socrates questions can stack that together to determine every single movement and thought you'll ever have? Thank you.
Okay. So, questions. All right, we'll start down here.
Thank you. I really enjoyed that. It was very interesting. This is probably a very biased question.
I was biased.
Okay. As someone that is interested in cell biology, so like at a very smaller scale, how do you think is best to combine like more macro brain mapping with like smaller scale, for example, cell biology, or even smaller, to like better understand the role of particular features and parts of the brain?
It's a fantastic question. It's one we're challenged with in my space often. And I touched on it a bit at the beginning, where you can appreciate it at least in the system it exists, or the circuit it exists. But you can argue more than that. The cell itself could be mapped. Every individual neuron is unique. There is no neuron that does the same thing as any other neuron. So should we somehow get to the point where we can map all of them, not just then, but we could then map how, we could then predict how the components of that cell dictate the neuron's behavior and then subsequently dictate the circuit's behavior and so on. So look, it's a while away. How do you implement it? Well, histology is probably the closest way at the moment, and immunohistochemistry, where you can look at how the cell then influences the nucleus itself.
Okay, we have a question up here.
Lovely.
Hi. I was just reading recently about Elon Musk.
Haven't we all been forced to do the same?
Yes. But putting the brain chips in these paraplegics, and I'm just curious whether he's used information that you've created or, mapped, and if so, does he pay for it? Or is it like a typical Australian discovery and we just give it away free? And do you see this must be a big
It is.
going forward for these poor unfortunates that end up as paraplegics and quadriplegics and everything else?
No, most definitely. It's an interesting field, the neurochip space, and it's got all sorts of other names. I don't like the fact that Elon Musk says he did it first, because there was a Swiss group that did it 3 years before him and a French group that did it 2 years before him. But that aside, yes, it is. Now, like an Australian invention, yes, these maps are more or less given away freely. Not entirely. The digital copies are given away freely, but if you want a physical copy, you will need to buy one. Not unfair, I think. And so they aren't used. At least Elon Musk did go to at least the Brodmann level, which I said is sort of the bare minimum medical level, and it shouldn't be something to be scoffed at. But he doesn't go any further. So 47 is still well under the 1,000 odd we have at the moment, but it is still more helpful than having none at all, and it's very, very empirically trained. Whether it's going to do all the science fiction we want it to do, it's not going to, but it is very quickly getting to the point where it's facilitating movement in people who, as you said, were paraplegic or quadriplegic, and you might have seen even recently there was an article with one of the case studies from the Swiss group where the girl who had a combination of a neural chip and then a prosthetic arm was able to control the prosthetic arm independent of her body, which is a nice direction for us to go. And I think we're getting to the point now with, if you wanted to look into the avatar system, yes, poor name, I know scientists are not very creative, which is being able to put chips not directly into the brain but into the spine, and being able to very quickly and safely facilitate movement in people who are not completely paralysed, but have damage to the spinal cord.
Thank you. We have another question here.
Yes, thank you. Very interesting talk. I was just wondering, do you map the brain of a particular individual, or is it an amalgam? And if it's one individual, do you try to select someone who's somehow typical?
It's a great question. So the convention in every other lab in the world is to take an average. So particularly for MRI studies, which is where most of the mapping is going now, the argument is you need a minimum of 1,000 healthy persons with no prior psychiatric disorder over the age of 25, under the age of 50. Now, that's hard to do, it's expensive to do, and it's very Americanized because they have the facilities to do so. But despite that, the maps made from our lab are of an individual, and there's a reason for that. It's not just cost, but by averaging everyone's brain together, it camouflages and washes out all the minute details. If I stacked an image of everyone's brain together, well, you're all going to see the big things. The cortical ribbon will be there more or less the same once we average it together, the thalamus, the hippocampus. But the small structures, the structures that are 2 millimeters big, well, they're going to get washed out and scrubbed away when you average them all together. And so our argument instead is everyone's brain is the same. Also, if you didn't know, everyone's brain is the same. Except how its circuits are combined. So, and on an anatomical level, we all have the same structures. So one healthy person scanned 100 times to get an amazing image is going to be more proof of what's inside our brain than taking 100 people and averaging all their instances of a scan and getting a very broad, sort of brushed image of what the brain would look like. Does that answer your question?
I think so, but I mean, do you try to select
yes,
someone
you do.
who doesn't have a particular sort of anomaly or something?
As best you can, but again, with us all having the same parts of the brain, unless it's a significant anomaly, which the radiographer will remove. I, for example, have a slightly larger sinus, which is one of the waste tubes in our brain, but that doesn't change the rest of my anatomy. I still have all the other bits, and I still have that sinus. And so by using the stereotaxic method, we can use the ratio of my map and then map that to yours, and the map coordinates will then be ratioed to match your dimensions.
Thank you for the talk, really enjoyed it. Can you talk to us about functional MRI? And the background of that question for skeptics particularly is that we get two classic areas, chiropractors and acupuncturists.
Oh, you must love them.
We love them.
We love them.
They put people in the MRI and they say, we place the needle here and this area lit up, or we manipulated the spine in this way and this area lit up. Can you talk us through what does lighting up mean?
Absolutely.
The
I'd love to.
Uses and abuses, basically, of fMRI.
Look, fMRI is helpful, and it has shown us some very broad things we didn't know otherwise. So fMRI, in its simplest terms, uses the BOLD method, which is the blood oxygen level dependent score, which measures how much blood oxygen a certain part of the brain is demanding. Under the assumption that brain activity uses blood oxygen to facilitate its activity, which it does, that score is then lit up on the screen representatively and argued that that's where activity is present. Now, the problem with that is that there's a delay of that score of about 10 seconds. Now, in those 10 seconds, a neuron can have 1 million activity points. So it's not just that, but also blood oxygen isn't the only measure of brain activity. And there is, in fact, my old supervisor would love this, Professor Bennett, who hated fMRI and spoke at the UN about how it's all bullshit and anyone who's using it is a liar. Because it can't be true. The brain doesn't just use oxygen. It uses so many other chemicals and so many other measures of activity. The best measure of activity at the moment is ECG. You know, putting all the little diodes you would have seen in movies even all over the brain. EEG, sorry, not ECG, that's the magic one. EEG. where that's actually measuring direct electrical activity as a result of neuronal activity. That's true. That is the neuron itself activating, being measured. But blood oxygen level is a correlated but not necessarily causative relationship to brain activity.
Thank you.
Okay, we'll have 2 more questions. So,
I can speak loud enough for everyone.
Yeah, you can.
Hello? Yes.
Oh, the battery died.
okay.
Hi, thanks for the talk, it's very interesting. I just wonder if you could tell us where we're at with understanding how memory works in the brain, because I know when you have a memory, it's all the senses, your smell, your memory, all those levels. So it's obviously a very complex process. Can you just give us, I know it's so complicated, probably hard to summarise, but can you give us a little bit of summary about where you're up to with how memory works actually in the physical brain?
We can. So, one sec. Oh, we can do that. That works too.
So just in case you didn't hear at the back, we wanted a quick summary of how memory works in the brain and where we're up to with the research in that space. It's an interesting little one, bit of history again. I know you're bored of it, surely. But we started with the engram approach, which is that memories are sort of encoded across the whole brain and you can't really pull them out. Dementia quickly proved that as a wrong idea because we can remove specific types of memory from by destroying certain parts of the brain. But how memory is formed, the quickest analogy would be a computer. So we have a working memory, similar to a RAM on your computer. This holds memories around you for about a minute. What's going on in and around you is quickly being stored and processed. What happens then is that it gets filtered from the hippocampus, which is where your working memory is done, through your insular and other cortexes in the ribbon to determine if it is of importance to keep. Now, unfortunately, the importance is almost exclusively emotional. It is not given from a logical standpoint. Smell, being the most evolutionarily conserved sense, has the strongest influence on our emotions, and that's why smell is usually the one that survives dementia the most, and usually why you can remember the scent of your mother's or grandmother's perfume. That then gets coded with emotion and gets stored in various parts of the brain. However, memory comes in 2 types, liquid and crystallized. Crystallized memory is your recall of exact facts. What's this road's name? What's the name of this noun? Where did I live? What's a 2-wheeled machine called, etc., etc. Where liquid memory is your ability to use things you have stored in memory to problem solve unconsciously. And so this is usually the more direct measure of intelligence when we do intelligence measuring, and it's unfortunately the thing that deteriorates the most quickly after the age of 25, which is your ability to use liquid memories to unconsciously problem solve. However, we can take some solace in the fact that crystallized memory doesn't deteriorate in a healthy person until about 110. So, yeah. That's sort of the quick summary. We could go into more, but that sort of covers the gamut, I think. There was one more?
One more question. Can you, just one more question.
Are we around if there's other questions? You can grill me, don't worry.
Are you able to shout?
Yeah.
You need the mic.
No, I can shout.
I know you can shout, but for the recording, it's
Oh, okay.
yeah.
Did you know your pick?
I had a couple of questions, so I'm going to have to choose which one. Okay, so I guess the more important question, which was slightly covered already, is you seem to fudge a little bit when you were talking about physical functions of the brain that you're observing and mapping, and then functionality of the brain in terms of driving behaviours. Can you talk us through how well is that mapping, if I can use that term, between physical, structures and behavioural functions these days?
Absolutely. In a moment.
Run, Jessica, run.
Thank you. A good question, because it's one that should be asked, that we have to be very cautious about. We can do it quite well, though. Not in the human, but we can do it extremely well in simple creatures, which gives us the understanding that it must be possible to do so with the same level of accuracy in the human. So in the human, we can go and take out a large chunk of your brain and see a large change in behaviour.
I prefer you didn't.
Yeah. But if we take out a small chunk of the human brain, it is harder or impossible to see such behavioural change. However, in simple nervous systems, such as those of the jellyfish, mosquito, and other rather simple central nervous systems, which, if they're under a certain number of cells, are called a neural net, because they don't quite reach the level of intelligence, we can manipulate individual neurons to get individual outcomes in those species of behaviour. So the argument should be the same in the human. But we have so many extra degrees of modulation in our behaviour that it's not that simple. So the last part of your brain to develop is the orbital frontal cortex at about 25. And that's sort of the governor general of your brain. You've got your cortex and your prefrontal cortex, and all your cognition and thoughts come from there. And that's great. We come up with great ideas and fun things to do. But at about 25, tiny bit younger for women, they get the orbital frontal cortex coming online. Just to be clear, it doesn't just magically appear. It was always there, but it becomes connected and active. And that goes, well, hang on, even though I want to go and get a can of Coke from the corner store that's over there across the road, I know how to do that, and I want it, and I know how to get there, and I've problem solved that I've got to cross the road and cross the steps. It's the orbital frontal cortex that goes, maybe you shouldn't cross while it's a green light, because you might get hit. And so that creates a lot of problems, where if we go and remove the part of your brain responsible for, say, one particular type of behaviour, your orbital frontal cortex might still very well be there to go, no, we're not changing our behaviour at all, because I can modulate the upstream effect and go, no. So the complexity gives us a lot of survivability as well, and that's why people with severe brain injuries can oftentimes not have much behavioural change and can be very functional members of society, because it's a very fail-safe system with all of these extra components, and that goes into neuroplasticity. But just on that, because it's something very cognate, is neurocriminality, right? Which is with this ability to dictate behaviour from understanding the function, the structural component and its dictation to behaviour. We were able to brain scan from early in the millennium, so from about 2006 to 2010, the ability to dictate those who are going to be more likely to commit a crime than those who would be not. So very minority report. And at the UN, Australia was actually the one that represented neuroscientists internationally, and said at the UN at a special conference that we should stop all neurocriminality research internationally, because it can only be abused. And so we are cognizant of the fact that structure and behaviour are linked to the point where it could be dangerous to continue to do research into it, because should it fall into the wrong hands, it would be abused.
Oh, sorry, I turned it off automatically. Force of habit.
Well, could everybody please join me in thanking Dr. Steve? And I know we didn't get through all the questions, and I know there are people who have questions, so
I'll be around.
He'll be around. He's still got dinner to eat, so please come and chat. Oh.
Thank you very much, everyone.
And everybody, take a magazine with you when you go.